topic I will touch only briefly because this will be the topic of the second speaker. Now let's start with the temperature level. So at which temperature we should keep our patients? Well, most of you will know the guidelines. These are the guidelines of the European Resuscitation Council from 2010. And 2010, the level of, it's okay, I can do it here. The level of temperature was recommended to be between 32 and 34 degrees. In 2015, the recommendation changed to have a temperature level between 32 and 36 degrees. Now, is anybody of you in the audience know why they changed the temperature level? You know. No? Anybody knows? So if nobody knows, that's good, so I have something to talk about. I will explain you why this temperature range changed. This is This is the temperature curve of the first randomized clinical trial back in 2000, which led to the initial recommendation of a temperature between 32 and 34 degrees. So here you can see the treatment, 34 degrees, and this is the control group. And if you look very carefully, you will see that the control group actually was slightly hypothermic. So they were not totally normothermic. And there were many voices saying that actually maybe it's enough to keep the temperature at normothermia, and maybe it's not necessary to cool down to 33 degrees, because what that study proved was that 33 degrees is better than slightly hypothermic. So this was the criticism. And that's why there was another study designed to investigate the difference between, I can use my mouse. Oh, there's no mouse. Thanks. So, one more. So slightly hyperthermic, 37.8 degree was this group. So that's why a new study was designed investigating 33 degrees, versus 36 degrees, so strict normothermia. And this was published in 2013. Now the outcome of this study was, this is the probability of survival, that between 36 degrees and 33 degrees, there was no difference in outcome. So that's why the new guidelines changed to a temperature range of 32 degrees up to 36 degrees. Now I would like to make the case why I still think we should cool the patients to 33 degrees, not going to 36 degrees. This is a very simplified slide, what happens after resuscitation from cardiac arrest. So there are many cascades leading to apoptosis or to cell death. It's very simplified. In reality, it's much more complicated. But it was shown in many cell cultures that a temperature of 32 to 34 degrees is beneficial on all of these cascades, or most of these cascades. There are no cell culture studies showing that 36 degrees is beneficial. And then there are numerous animal studies showing a benefit of 32 to 34 degrees versus normothermia. Countless. I put 50 of them. So they all uniformly show the benefit of 33 versus normothermia. And this is one of these studies. I want to go in more detail because it shows very nicely the U-shaped effect of temperature. This is a rat study in cardiac arrest, and here you can see different temperatures. Red is the normothermia, yellow is 35 degrees, green is 33.5 degrees, and blue is 30 degrees body temperature. And these are the number of damaged cells in the brain. And here you can see the best effect is 33.5 degrees. So 33.5 degrees has more effect than 35 degrees, more effect than normothermia, and more effect than 30 degrees. So it's a U-shaped effect of temperature. And that's just another parameter, caspase 3, a parameter for cell damage, and 33.5 has the best effect. And the third, so it's the other way around, the more the better. Again, 33.5 degrees has the best effect as compared to 35 normothermia or 30 degrees. So U-shaped effect of the temperature. And this is a very old study, but a very interesting study, because it investigated different temperatures before cardiac arrest, during cardiac arrest, and after cardiac arrest. And I picked two groups. One group is 36 degrees throughout the experiment, and the other group is 36 degrees before the cardiac arrest and 33 degrees after cardiac arrest. The ones of you who listen very carefully to my talk will recognize that that's exactly the temperature of this trial, which showed in humans no difference in outcome. But in animals, nicely controlled study, there is a benefit of 33 degrees versus 36 degrees expressed in the amount of dead cells. So in animal studies, there is a benefit of 33 degrees versus 36 degrees. This is a complicated slide, and I will guide you through. That's a meta-analysis of all randomized trials with target temperature management. So these are the studies, and that's the result. And if this line does not cross this line, it's statistically significant. So this study, statistically significant, significant. This is borderline significant, the Bernard study. But all in all, there's a benefit of 33 degrees versus normothermia. Now, if you add now this study, this is the Nielsen study, showing no benefit between 33 and 36 degrees. If you put all the studies together here, there is a statistically significant benefit of 33 degrees versus normothermia, including the study which led to the recommendations to have a temperature range of 32 to 36 degrees. Now I will show you what happened in Australia after implementation of the guidelines when the hospital changed from 33 degrees to 36 degrees. So there were 24 patients before the new guidelines, 33 degrees, and 52 patients after they changed the temperature. So what do you think happened? Do you think the outcome remained the same? Do you think the outcome became better or the outcome became worse? What do you think? Well, there's the answer. The answer is, in blue it's the 33 degrees, in red is the 36 degrees, and you can see that hospital survival, good neurologic outcome, CPC is cerebral performance category, and discharged home, less patients were discharged home, so outcome became worse after changing to 36 degrees. Now this is a new study from 2018, unfortunately it's cut off here, so it's from 2018, and that's too fast. The next one. Oh, it's missing. So, this study shows from Australia and New Zealand 16,000 patients from 2005 to 2013, the temperature curve. So the temperature during the years, it got not to 33 degrees, which should be the target temperature, but to 34 degrees. And then here, the new guidelines, and most of the hospitals changed to the new temperature range, 36 degrees. This is the temperature. Now, if you look at the outcome, mortality, that's the mortality, went down over the years, but when they changed to 36 degrees, mortality started to go up again. So, taking all the evidence together, I'm convinced that 33 degrees is the right way to go, and I show you one slide which might explain why 36 degrees is not that good. Here you can see different temperatures measured in the rectum, esophagus, as compared to brain temperature. And most of the time, brain temperature is higher than the temperature on the other sides of the body. So meaning body temperature or brain temperature is up to 2 degrees, 3 degrees higher as body temperature. So if you keep the body at 36 degrees, it could be that the brain is 38 or even 39 degrees. And hypothermia is bad for the brain, that we know for sure. So, taking together all the evidence, I would recommend, and many experts in the field still recommend, to use the 33 degrees. Now the next question is, when to start cooling? I think you have the wrong slides for the translation, right? You don't have the same set of slides.
Maybe.
Okay, when should we start cooling? If you know the recommendations from 2015 from the European Resuscitation Council, it says that pre-hospital cooling using a rapid infusion of large volume of cold intravenous fluid immediately after restoration of spontaneous circulation is not recommended. So they say don't cool out of the hospital. But if you remember, oh, that's the right one. More, okay. More, long, okay, more, one more, one more. Okay, here we are. Thanks.